Alright, cell cycle and cell division. When there is a damaged, dead and worn cells, it can be replaced by growth and divisions of similar cells. And each of the cells inside your body, had been programmed. We call it the self-destructions, which means that every single cell inside your body had been programmed, and then they will know how long is their lifespan. For example, your blood cell. Alright, the red blood cells, the lifespans start from it was being formed in your in the bone. Alright, so the blood cells have the lifespans about 120 days, which means at the end of the 120, self-destruction occurs where the red blood cells will be self-destruct and it will be removed by the process of phagocytosis, which is known as apoptosis. Alright, look at this picture again. Okay, this is the process. The green one, it is an interphase, which is the process when there is no mitosis happen. It only the process of cell growth. Okay, this is the interphase, is the process where it takes the longest time before it can be divided. Okay, the red one is a process of mitosis. This phase, they call it M phase, where the cell divisions occurs. Okay, and then it will divide. In the process, we call it cytokinesis. Okay, once it is divided, right, each and individual cells will go through the phase of G1 phase. At this phase, the cell will be growth. Okay, will be given, go at this state until it is really matured enough, which is have the sufficient nutrient and the sufficiency of oxygen. Then only the cell, it is fit for the DNA replication. When there is a process of DNA replication, it already go to the S phase. Right? And then when they are ready, before the process of mitosis begin, the cell growth continue. Then only it will enter the post-process of mitosis, which can be further divided into 4 phases. Right? So interphase, on the other hand, it was called the preparations of cell division. It is a period of non-divisions. It is a phase where all the DNA replicates, and during the replications, if there is any mistakes occur, it is need to be corrected immediately. So the correction was done by the enzyme, and if it is not corrected, the mutations occur. Okay, so this is the part where the centrioles also divide, and then the proteins are actively produced, and there is an increase of metabolic activities, whereas all the creating of energy and producing waste products happens. So this is the phase where the cell divisions is preparing for the cell growth before it can go further to the next phases of mitosis. So interphase consists of 3 parts, or they call it 3 checkpoints. Checkpoint is a critical control point with stop and go ahead signal that regulate the cell cycle. Do you remember just now I said the cell division, The cell cycle is a controlled process, right? Interphase is the longest phase of all. Divide into 3 checkpoints: G1, S phase, and G2 phase. G1 restriction point. This is the phase where the cell have to be large enough to divide. It needs to have a sufficient nutrients. When there is a sufficient in nutrients and the cells is matured enough, it will receive a signal said go ahead. When go ahead, it will go enter the S phase. If cell did not receive a go ahead signal, it will exit the cell cycle and switch to the G0 phase, a non-dividing state. S phase is the process of DNA synthesis, where there is a DNA replications was done. And G2 further growth and readily to go to the M phase. This is the summary of the process of mitosis. Okay, after it go to the G2 process, it will enter the M phase, which consists of 4 subphases. And this mitosis is only can be seen in the somatic cells, which is all the non-reproductive cells. So the first phase is the prophase. The second one is metaphase. The third one is anaphase. And the last one is the telophase, and followed by the cytokinesis. So mitosis can be defined as a single cell. The end product, it will produce a 2 identical daughter. Means the daughter is identically or exactly like the parent's daughter. The parents. It have the same number of chromosomes as does the parent cells. Okay, this is the picture of the prophase. At this phase, the chromatin condense. The spindle fibers form, right? Did you see? This is the spindle fiber. And then the centrosome migrate. Okay, it will migrate to the pole. And it will go to the next phase, which is metaphase. The microtubules will attach to the chromosome, and the chromosome move to the midline plate. This is the spindle, this is the polar split fiber, and this is the chromosome. This is the spindle form, pole. After it goes to the second phase of metaphase, okay, the third phase, they call it anaphase. At this point, the link will break. You remember just now it line up here, all right, all the chromosomes line up, okay. The link breaks, and then the 2 daughters of chromosomes move to the poles. The process of movement, it is a process, means that it will go to the anaphase subphases. Okay, before the last process of cell division, okay, the chromosome, at the end, it will reach the poles and decondense back. And then slowly the nuclear envelopes reform and the spindle fiber disappear. Now, this is the last part of the cell division. When there is a cleavage of forest and also microfilaments, the cytokinesis begin. The cell membranes will pinch inward like a belt, tightening between the 2 poles until the 2 daughters are formed.